Okay, so now we're going to be talking about genetics and heredity. So hello everyone, my name is Michael Zhao. I'm the founder and president of Silicon Valley STEM for Youth. And so I've been doing life science competitive events for a very long time, starting from middle school. I'm in the top 50 of the USA Biology Olympiad, and I've been doing lots of life science Olympiad events, disease detectives, designer genes, which our presentation today is partially based off of, cell biology, protein modeling, microemission ecology, anatomy, and experimental design. So lots of life science going on here. I'm the founder and president of Lynbrook High School Science Olympiad Club as well. And I was on the 2017 National Science Bowl Middle School Championship team. So I'm hoping that through today's lesson, we can get into the fundamentals of genetics, why it's so important, and just the things that you'll need to know to succeed in the genetics events in Science Olympiad, which are designer genes and heredity at the middle school level. So let us begin. Genetics is the study of heredity, and I think a lot of you have probably heard about genetics in one way or another, whether it's through 23andMe and the genetic testing kits, whether it's through just seeing this molecule of DNA. A lot of people know about the double helix starting from elementary school, or just looking at your parents and your siblings and wondering, why do I look so similar to them? And so our ancestors knew about heredity from very, very long ago, and they were always wondering about why. Why is there heredity, and what controls it? So for this presentation, I'm going to define kind of two sections of heredity that we're going to want to look at. The classical genetics, Mendelian genetics. If you don't know who Mendel is, we can get into that first. And molecular genetics, and the actual molecules, the DNA that is behind inheritance. But as you'll see very shortly, these two are actually quite closely intertwined. So let's start with our favorite Austrian monk. His name is Gregor Mendel. And Gregor Mendel, he was growing a pea garden because he wanted to learn about how heredity, how inheritance works. And so if you haven't seen one before, here's a pea plant. Notice that you have like the pea pod, you have flowers, and they have a certain color. This will become important later. And so, as it says here, Gregor Mendel was a pea garden farmer, and he discovered some of the fundamental laws of genetics through his experiments in his pea garden. And so here are some of the traits he observed with his pea plants. He saw that for the pea plants, there were certain characters that had different variants. For example, some of the pea plants were tall, some of them were short, some of them had purple flowers, some of them had white flowers, some of them had yellow peas, and some of them had green peas. And these were very rigidly defined. Like he only ever saw these two variants of characters on his pea plants. So just take a look at them really quickly. You don't need to know all of them, but it's just like height, flower color, the flower position, seed color, seed shape, the color of the pea pod, and the shape of the pea pod. And so let's focus on one of them. So just flower color for now. So Gregor Mendel, he wanted to try breeding these flowers with each other to try to determine how heredity worked. And so he got a true breeding purple flower, which means that it's a purple flower that if you breed it with other purple flowers, all its descendants are always purple. And he got a true breeding white flowered pea plant, same concept, but for white flowers. And he found that if he cross-fertilized them, like with the pollen, then the descendants of this cross, of this mating, all had purple flowers. So this is very interesting, right? Because one of the parents had white flowers, one of them had purple flowers, but its descendant, the F1 generation, P stands for parental generation, the F1 generation only had purple flowers. Kind of like the white flowers just kind of disappeared. But he went one step further. This is where he discovered a lot about what we know about fundamentals of genetics. He fertilized the purple flowers, the F1 flowers, with themselves. And this is something you can really only do for plants. So self-fertilization, and he discovered that they fit this very nice, very mathematical 3-to-1 ratio of purple flowers to white flowers in the descendants of this F1 cross. So again, to elaborate, he had these parental genotypes, he had phenotypes. He had a parental purple flower and a white flower, and these were true breeding. And he crossed them, and the next generation, all the flowers were purple. But when he crossed this generation of purple flowers with itself, they got a 3-to-1 ratio of purple flowers to white flowers. So he wanted to investigate this really nice mathematical ratio. Usually means that there's some kind of law at work, some kind of natural principle you can quantify. So he defined a few important words. And I say that as quite an understatement. They are very important. The first one is genes. So genes control traits like the flower color. And they come in a pair of different alleles. So as you can see here, alleles are different variants of a gene. And so, for example, he would call the one that caused the flower to be purple uppercase B or big B, and the one that causes white flowers little b or lowercase b. And he called the combination of alleles that each individual pea plant had the genotype. So, for example, this genotype is big B, big B. This genotype is little b, and he called the observable trait of the individual the phenotype. So the phenotype of this flower is purple, the phenotype of this flower is white. And so again, genes are units of inheritance. And so genes control certain traits. The gene that he's looking at here is the flower color gene. It has two alleles, the purple allele, big B, and the white allele, little b. They each have different genotypes, these two flowers. This one has a genotype of big B, big B. This one has a genotype of little b. And the phenotypes are purple and white. And then he defined a few of the what are known as Mendel's laws now. The first one that we're going to talk about is the law of segregation. So this nomenclature that I just went over with genes, alleles, all that stuff, that's all very helpful for understanding his laws. So the first one you'll probably want to know about law of segregation says that each offspring will only inherit one allele from each gene from each parent. So this F1 generation, the children of this P generation cross, the F1 gets a big B allele from its purple parent, right? And it gets a small b allele from its white flower parent. And so the F1 will have a genotype or this allelic combination of big B, little b. It got one of its alleles from one of its parents. It got another of its alleles from another parent. So that's the genotype of this F1 generation flower, big B, little b. The next law is the law of dominance. And so Mendel noticed that when you have, for example, in this F1 generation, you see that has both the big B and the little b allele. And we said that the big B allele causes the flower to turn purple. The little b allele causes the flower to turn white. But as you can see, the F1 generation is completely purple, even though it has both of these. So Mendel saw that this big B allele, the reason why we write it big, is because it was dominant to the little b allele. That is, when an individual has this dominant allele, this is what it expresses. So if this pea plant has a genotype of big B, little b, or big B and big B, it will always show the purple dominant trait. And it will only show this recessive, we call the allele that's not dominant recessive. They only show this recessive trait when it has both these recessive alleles. So when the dominant allele and the recessive allele are together, the dominant allele, big B, dominates the recessive allele, little b, and what it shows is the trait of the dominant allele, which is the purple flower coloration. So that's why F1 is always purple. So it gets the alleles from its parents, one from each parent, and then it has this genotype over here with big B, little b, and because of the law of dominance, it always looks purple. And then so here's just a quick terminology review. This is very, very important for understanding genetics, which is why we're spending a bit of time on it. So the dominant allele is always expressed over the recessive allele. This purple allele is always expressed over this white allele if they both exist together. And it's expressed by this capital letter. We use capital letters to denote which allele is dominant. And it's called homozygous. Homo means the same. Homozygous if both alleles are like this, big B, and it's called heterozygous if it is big B and little b. Hetero means different. So heterozygous if it's big B, little b. And hetero means different, so it has two different alleles. But of course, as we talked about the law of dominance, it expresses the dominant purple trait instead. And the recessive is masked by the dominant. That's the white little b. It's written in lowercase letters, so we know that it's recessive. And it only shows if the pea plant has a homozygous recessive genotype, or the combination of alleles is just both lowercase letters. And then one way we can find these crosses, like what the descent will look like, is by using a Punnett square. And so that's a box showing all these allele combinations. So we talked about the law of segregation. So you take, this is the F1 cross, the F1 crossed with itself. You have the F1 flower, it has one big B allele, one little b allele, and these are segregated by the law of segregation. So we consider them like this. Same thing for the female parent, which is actually the same flower. And then you just cross them together. Big B gives you two big B's. Big B, little b gives you one big B, one little b. Little b, big B gives you one big B, one little b. And little b gives you two little b's, so homozygous recessive. And as you can see, you end up with one homozygous dominant flower, two heterozygous flowers, and one homozygous recessive flower. And as we talked about with the law of dominance, the homozygous dominant and the heterozygous flowers are all purple. And this flower here that's homozygous recessive is white. And so that's what Mendel saw in his 3-to-1 ratio. So now we know where this 3-to-1 ratio is coming from mathematically. And so what we just performed, sorry, let me erase the annotations. What we just performed is called a monohybrid cross. Mono meaning one, hybrid meaning they're different alleles, because you have this big B, little b crossed with big B, little b. So this F1 cross producing the F2 generation. And so that's one gene or trait, for example, flower color gene, which is the purple versus the white trait. And then you get a Punnett score that has 4 boxes. And this is the monohybrid cross. And as you can see, it always produces this 3-to-1 phenotypic ratio of what we can observe, and this 1-to-2-to-1 homozygous dominant to heterozygous to homozygous recessive genotype frequency. So yet again, you can see it summarized here. And so now we're just going to check in every once in a while, make sure everyone is getting the concepts we're talking about. So we're going to have a quick quiz time. And I'm going to put up a poll in the Zoom. So the question is, what is the genotypic frequency, the genotype frequency resulting from a monohybrid cross? So remember, we talked about two frequencies, the genotype and the phenotype. So you're going to have to distinguish which one is which. Okay, I'll give about 10 more seconds. So it's a 1-minute poll. Okay, great. So we got a majority of people getting the correct answer, but let's go back and review really quickly. So the genotype frequency is 1-to-2-to-1. So the genotype again is this arrangement of letters, these alleles that each individual has. So we have one homozygous dominant, two heterozygotes, and one homozygous recessive. And in a regular Mendelian monohybrid cross, this is always the genotypic frequency you'll express. So the genotype is the actual genes, the alleles that the individual has. Whereas the phenotype is what you can observe. So the phenotype is purple or white. It's what we can see. We can't actually see the difference between this homozygous dominant pea plant and these heterozygous pea plants. Notice how they're all purple, even though one of them is homozygous dominant, one of them is heterozygous. So phenotypically, they're the same. But genotypically, they're different. And that's why this genotype frequency of 1-to-2-to-1, we know the homozygous dominant and the heterozygous, they have the same phenotype. So you add 1 and 2 together, and that's how you got the phenotype frequency of 3-to-1. Okay, so now let's look back at another of Mendel's observations, and that is going to be the pea pod, the actual peas, the seed color. So here's the character he's looking at, color and shape, and there are the variants. He has yellow peas, sometimes green peas, sometimes he has round peas or wrinkled peas. And so this is what we're going to do. What we're going to do is called a dihybrid cross. So what we just talked about are monohybrid crosses, right? Where you have this one gene for flower color, and then you just cross them together and try to find out what the offspring were. This time you have a dihybrid cross. Di means two. So you have two genes, seed color, seed shape. And we're going to try to find out essentially what are the genotype and phenotype frequencies for this dihybrid cross. So again, monohybrid cross, we had a 3-to-1 phenotypic frequency, a 1-to-2-to-1 genotypic frequency, but it's different for a dihybrid cross. So this is where Mendel got his third law, the law of independent assortment. So each gene in its alleles, he said, are inherited independently of each other's genes and their alleles. So the seed color gene and how it's inherited is completely separate from the seed shape gene and how it's inherited. There is no connection or relationship between these two. So it's almost like we can consider these two genes to be inherited separately or independently, which is why he's calling it independent assortment. So the inheritance of seed color and seed shape do not affect each other. So that's why we're going to continue to follow Mendel's law of segregation. In essence, what we have here is it's just two monohybrid crosses on top of each other. So we're going to take one allele from each gene of the parent, right? So the parent here is, if we just look at this seed shape, we have big S and little s. We're going to take one allele from each gene of the parent. So big S, when we take little s, and same thing for this other parent, big S and little s. And we're going to take one allele from the other gene we're considering, which is the seed color. So that's YY here. So we take big Y and little y, and big Y and little y. And so as you can see now, we have the four combinations of independently assorting alleles. Big S, big Y, big S, little S, big Y, and little s, little y. And these are all coming from this one parent, and it's independently assorted. So because we used to have only two choices between each of the genes alleles, and now we have 2 times 2, so we have 4 choices. So that's why we have these 4 on the axes. And so if we cross them together just like we did with the other Punnett square, you get all these genotypes. And as you can see, the genotypes are different sometimes, the phenotypes are different sometimes. And what you end up seeing is in this dihybrid cross, where we have 2 traits, you have a Punnett square with 16 boxes. So remember the monohybrid cross, we had 4 boxes, now we have 16 boxes, and 16 just happens to be 4 squared because we have 4 to the power of 2. We have 2 different genes we're considering. Ignore the genotype ratio right now. This is just for fun. You'll notice the genotype ratio is a bit of a tongue twister, 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. But the phenotype ratio is what they normally ask about. So phenotype ratio here, we count for these smooth yellow peas. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 smooth yellow peas. We have 3 of these smooth green peas, 1, 2, 3. 3 of these yellow wrinkled peas, 1, 2, 3, and just 1 green wrinkled pea. So that's the phenotypic frequency that we're getting from this dihybrid cross. And you can calculate that, again, by drawing this Punnett square, independently assorting each of the genes, kind of like pretending, first you pretend that seed color doesn't exist, and then you pretend that seed shape doesn't exist when you're sorting out these alleles. And then you show the Punnett square, and you can count the phenotypes and the genotypes.